Greetings everyone. I am Krishnan, a graduate student in Biomedical Engineering Department at Duquesne University. Today, I'll present our work on developing a robust microcontact printing platform for high-precision cellular pattern. Why cellular patterning? Cell-cell interactions are fundamental to life, governing processes from embryonic development to disease progression. To understand the forces in mechanobiology, we need precise control over these interactions. We use microcontact printing to create defined adhesive islands, cells preferentially attached to these pattern areas, allowing us to dictate their organization and study specific pair interactions. So the first image shows the fluorescent image of a fibronectin structure in pattern design, and the second one is our goal of forming perfect pairs. Microcontact printing protocol is performed through a sequential process beginning with the fabrication of pattern PDMS stamp via soft UV lithography. This stamp is subsequently cut into small pieces and incubated with a fibronectin solution for an hour. Following incubation, any unbound protein is removed by washing the stamp with deionized water and dry. The final steps involve stamping this fibronectin pattern onto the surface of a 6-well plate and then passivating the remaining non-patterned areas by coating the entire surface with bovine serum albumin to prevent non-specific cell adhesion. Following patterning, HEC293T cyclide cells are seeded at a concentration of 0.3 million cells per ml. After a 20-minute initial attachment period inside an incubator, unattached cells are removed and replaced with fresh DMEM and incubated further. Time-lapse fluorescent imaging is performed at 6, 14, and 20 hours, and the results are quantified. We systematically varied 3 key parameters to maximize patterning efficiency. Firstly, we tested 3 fibronectin concentration 20, 30, and 40 microgram per ml. A concentration of 30 microgram per ml yielded the highest density of on-pattern cells. Cell viability remained high and constant across all 3 conditions, indicating concentrations were non-toxic. Secondly, we tested various BSA concentrations, 500, 1000, and 1200 microgram per ml for preventing non-specific attachment and identify the optimal level of passivation. 1000 microgram per ml BSA resulted in the lowest level of off-pattern cell attachment, forcing cells to adhere only to the destined pattern. Cell viability for the on-pattern cell remained consistently high across all concentrations. Finally, trypsinization cleaves cell surface off of adhesion proteins. We investigated the recovery time required for cells to regenerate these receptors and restore specific adhesion by incubating them in DMEM for varying periods of post-trypsinization. 2 hours, 4 hours, and 5 hours were examined. A 4-hour recovery period in DMEM yielded the highest patterning efficiency. Again, cell viability was consistent, confirming extended time in suspension had no adverse effect. To show how we define cell pairs, the figure A shows exactly 2 cells or as a cluster of 2 cells. These are pairs, and the figure B are non-pairs having 1 cell or more than 1 cells, and figure C shows off-pattern cells. To analyze the cell pairing efficiency, the occurred images were analyzed to quantify cell attachment, distinguishing between on-pattern and off-pattern localization. Cells were categorized based on cluster size, ranging from single cells to pairs and larger clusters up to 8 cells. The number of cells forming perfect pairs versus non-pairs was specifically determined. The cell clusters evolved over time, showing proliferation and cell health as shown in the figure B. To conclude, this work establishes a robust microcontact printing platform for high-resolution adherent cell pairing, achieving controlled patterning with over 40% pairing precision. Key optimized parameters were identified. A fibronectin concentration of 30 microgram per ml proved optimal for HEC293T cyclide cells, while a BSA concentration of 1000 microgram per ml effectively passivated non-pattern areas. Furthermore, a post-trypsinization rest period exceeding 4 hours was critical for cells to regenerate adhesion receptors, maximizing patterning efficiency. That concludes my presentation, and thank you